Memories are an important part of our lives. Memories, in essence, are the only thing that we take to our deathbed.
The hippocampus, which is Latin for seahorse, is a small organ within the brain's medial temporal lobe. The medial temporal lobe forms an important part of the limbic system, which is the region that regulates emotion. The hippocampus in particular is mainly associated with memory. The lateral occipital cortex is a general-purpose object processing region focusing on an object's shape, size, and location.
Previous animal studies suggest that hippocampal signals have a link to remembering events across temporal gaps. What does that mean? The way we judge the length of this gap is what we refer to as the retrieval of temporal proximity between two events. In previous human fMRI studies, it has been shown that the hippocampus is activated when people have been asked to bind together elements that are either spatially or temporally separated. The caveat of this study was that it was a single trial and can't significantly represent the hippocampal link to temporal memory.
The nature in which the hippocampus contributes to memory-related binding of events in a reasonably sized sample group has not yet been investigated. Moreover, it's also unknown how the physical context of where a memory is encoded may influence the hippocampal mechanisms of associative memory across time. The present study aims to address this gap in literature by investigating whether physical context would have any impact on the judgment of temporal proximity across different events that are temporally separated. That is, whether the setting in which the memory of events are formed has any impact on your judgment of how temporally close those events were. Moreover, the study aims to investigate how stability in the pattern of hippocampal activation across trials is related to temporal memory for information encountered during those same trials. That is, whether the activated hippocampal bold signal would have any correlation to the bold signals we observed during the context condition in temporal memory trials.
If this research question is addressed effectively, we will have a better insight into the encoding and retrieval of memories that are temporally separated. The important implications of this being addressed is that it may help us to assist patients who are suffering from retrograde and anterograde amnesia, and also other conditions that involve the impairment of memory. This study is interesting because it's the first of its kind to use a sample size larger than 1, and it provides evidence towards the encoding and retrieval of temporally separated events and its link to the hippocampus.
Researchers used 21 right-handed participants who were native English speakers aged 18 to 31 from the NYU and NYC surrounding area. 13 of them were female and the other 8 were male. Participants performed an associative encoding task on each trial. Stimuli for encoding and retrieval tasks were nameable objects and famous faces taken from an online database and internet search, and outdoor scene stimuli were taken from just an online database. Stimuli lists were created for each participant from 128 objects, 128 faces, and 96 scene stimuli. No objects or faces were paired with the same scene more than once across participants, and all stimuli were presented equally often in the context boundary and same context conditions. Scene stimuli repeated across either 2 or 4 consecutive trials, while object and face stimuli were trial unique. Objects and faces that were separated by a change in the associated scene made up the context boundary condition, while objects and faces that were paired with the same scene made up the same context condition. Participants were presented with an outdoor scene paired with an object or famous face for 4 seconds and were instructed to imagine a scenario where the person or object was in the scene. Participants went through 64 encoding trials per run for a total of 4 runs overall, and after each stimulus, a screen appeared that cued them to make a yes or no judgment on their imagery success.
Following each encoding block, participants performed a memory test in which they were asked to rate the temporal proximity between the pairs of the items from the encoding block. Participants made their responses on a 4-point scale: very close, close, far, and very far. For the purposes of the behavioral and fMRI analyses, responses were collapsed into 2 bins: close and far. Temporal memory was analysed based on whether the item pairs were from the context boundary or the same context conditions during encoding.
Following a 10-minute delay after the scan session, participants performed a surprise source memory test for the associations between the trial-unique faces and objects and their paired scene stimuli. Participants were presented on each trial with a face or object from encoding and 2 alternatives for the associated scene stimulus. Participants chose either the left or right scene stimulus while indicating high or low confidence in their choice.
This figure shows the mean proportion of pairs that participants labeled close compared to far. Participants were more likely to rate the same context pairs as close compared to the context boundary pairs. Within the context condition, participants were also more likely to rate pairs as close compared to far. This figure shows the mean response times to label pairs as close and far. For the pairs labeled close, participants were slower to label context boundary pairs compared to the same context pairs.
Pattern similarity for pairs of items labeled close was higher than for pairs labeled far in the left hippocampus. Left hippocampal pattern similarity was enhanced for close pairs relative to far pairs in the context boundary condition. Pattern similarity was also enhanced for close pairs compared to far pairs in the left occipital cortex. In contrast to the left hippocampus, pattern similarity in the left occipital cortex differentially predicted close-far memory in the same context condition, leading to a region X context condition X memory response interaction.
A whole brain searchlight analysis revealed the number of regions, such as the right interparietal sulcus, in which pattern similarity was enhanced for context boundary pairs compared to same context pairs. The searchlight analysis identified regions such as the right medial prefrontal cortex, in which pattern similarity was enhanced for same context pairs relative to context boundary pairs. Critically, the searchlight analysis identified no regions in which overall pattern similarity was related to later temporal memory.
Yo, that's a great study. I'm gonna let you finish, but is a sample size of 18 really enough to significantly infer information about a whole population? And by using the region of interest method, could there be some areas that are less robust that were overlooked?
Those are definitely some limitations to our study.
Kanye does make some valid points. Although the region of interest suggests a hippocampal link to memory-related judgments for temporal proximity, a causal relationship can't be established from these results alone. Moreover, a caveat of using region of interest is that some areas that are less robust and consistent may be overlooked. Therefore, there may be other areas that play a role in the retrieval process. The sample size was originally 21 and then reduced to 18 due to a sleepy participant and head movement artifacts in the other two.
As we mentioned earlier, these results are important because it's a first study to use a sample size greater than 1 human to provide evidence of the hippocampal link to context-dependent temporal judgments. Looking at this on a larger scale, as our life expectancy increases, so do the complaints of memory failure among clinics and hospitals. Consequently, the need to improve memory and reduce the anxiety associated with its loss represents a significant issue among society. Retrograde memory is essentially our cerebral journey containing all our life's experiences, and anterograde memory helps us to form new memory and new skills. The loss of these functions may not only condemn you to a timeless limbo, but affects your ability to navigate your environment and your whole perception of the world. By adding to the body of literature surrounding memory and how it's formed, we're essentially working towards reducing the amount of memory-related disorders in society.